For this standard of lifespan development, I'm going to be highlighting the stages of embryonic development, which covers about the first 8 weeks after fertilization, but does not begin to talk about any fetal development. You'll notice most of the internal structures form here. They're still just very small, and they begin to really differentiate themselves as human-like tissue. The standard reads, I can describe the embryonic stage of prenatal development and includes birth defects. So what's happening in embryonic development? Well, when we say pregnancy, we're talking about there being a presence of developing offspring in the uterus. We divide a pregnancy up into 3 trimesters, each of which is about 3 months long. So first trimester, second trimester, third trimester. Most of the time, the average human pregnancy will last about 38 weeks from that initial moment of conception all the way through birth, though it's not uncommon to go early or to go into labor later. We divide this pregnancy up into 2 major stages, the embryonic stage, which is the first 8 weeks, and the fetal stage. Today, I'm highlighting the embryonic stage. So, during the embryonic stage, these first 8 weeks, we're forming a placenta. We're developing most of our main internal organs. We're seeing external body structures appear. So this is 1 original cell turning into 2 to 4 to 8, and then we're watching as those cells not only keep dividing, but start forming very distinct features. Step 1 is called the period of cleavage. So cleavage refers to really quick cell division that's taking that initial zygote cytoplasm and dividing it into smaller and smaller cells. When it's a small collection of undifferentiated cells, it's referred to as a blastomere. So to list a whole bunch of the staged names, the zygote was our fertilized egg. When we had 16 cells, more or less, it's still a solid ball and they're just dividing rapidly. It's floating in the fallopian tube. It has not yet embedded in the uterus. And at that point, it's called a morula. Eventually, that morula will hollow out and will have an outer lining and a little clump of cells on the inside. That hollow ball is the one that's going to implant itself in the uterine lining, which will then become the womb. The inner cell mass are those cells inside the hollow ball that will form the embryo itself, and that outer lining is called the trophoblast, and it will form accessory structures like the placenta and the umbilical cord. The placenta, if you're unaware, is a highly vascular structure, meaning there's a lot of blood vessels, and it provides an exchange of blood between fetus and mother, and that allows the fetus to get oxygen because it can't breathe yet. It allows it to remove its waste through the mother's blood and allows it to receive nutrients from mom as well. Around the 6th day after fertilization is when this blastocyst will finally adhere to the wall of the uterus and really begin to divide and grow into it. We'll have this fusion of cells where the zygote cells, the blastocyst cells rather, will start to grow down into the lining of the uterus. Step 2 is this implantation, when the blastocyst nestles in, and it occurs about 7 to 10 or so days after fertilization. So the first majority of divisions, while it's a solid ball and then while it hollows out, it's still just floating in the woman's body headed towards the uterus. Implantation occurs days 7 through 10. In step 3, we're forming some of the extra embryonic membranes, and we're starting to also form the placenta. So the chorion is a name for this outermost layer of what was once the blastula. It's made up of those trophoblast cells and the cells that are lining it, and they start to grow these slender projections that will erode into the endometrium and start to link up those blood vessels for blood exchange. Couple terms here, other things we're developing during stage 3. I'm going to focus on the final 4 here. I'm going to skip lacuna. So the amnion is a membrane that will hold all of the fluid that surrounds the baby. And that fluid is called amniotic fluid. This fluid protects and cushions our embryo. It also helps regulate internal temperature. So when a woman's water breaks, what you're seeing break is that amnion membrane, and then the fluid that comes out is the amniotic fluid. At this time, the umbilical cord is going to form from those projections that grew into the endometrium, into the uterus. So we have a connection now between embryo and mother through this umbilical cord. And then, of course, the placenta, which is a large, flat, pancake-like organ that just really acts to exchange surface area and exchange a lot of blood then between mother and baby. So it's like this entire placenta is attached to mom, but it funnels all those nutrients into the umbilical cord to go into the stomach of the baby. And that's what we're seeing here. So we have a giant placental surface, so a big area that's connecting to mom, but all those blood vessels will funnel into one umbilical cord that will deliver that blood to the baby. We see in blue here, we have our amniotic fluid surrounded by an amniotic sac, keeping that baby protected and cushioned. Step 4 is called gastrulation and organogenesis. So gastrulation is when we start to see cells form distinct layers. And we have 3 primary layers. The endoderm is our inner layer, and the ectoderm is our outer layer. This is going to form all of our epithelial cells. So it's going to form our skin on the outside. It's going to form any coverings like the pericardium, which was a covering of our heart, or the linings of our digestive system. Anything like that that's part of our epithelial tissue is formed by one of these 2 layers. Of course, ectoderm is more for skin, and endoderm might be more for digestive tract, but they will nonetheless form epithelial cells. Mesoderm will form connective tissue. So that is our muscles, that is our cardiovascular system, our bones. All of these things will form by the middle layer of mesoderm. While those layers are forming, they also can start the process of organogenesis, where they're creating distinct organs from those 3 primary germ layers. During week 4 or so, halfway through the embryonic stage, we now have this placenta, we have the umbilical cord, and we're forming the spinal cord, we're forming limb buds, head, jaws, and we even have a preliminary heartbeat. We have a little node that's beginning to circulate. And so we're starting to see not just a ball of cells anymore, but something that looks like it could be a reptile or a bird or a human. Because we can see those vertebrae in that spinal cord right there. In weeks 5 through 7, we see a lot of growth. So we see head growth, we see limb growth, and we're establishing most of our main internal organs, our liver, our stomach, etc. In week 8, the embryonic stage concludes, and we have something that is very human-like. It has all of the limbs and fingers and toes. It has most of our structures. They're just not fully developed. The last thing I want to mention is the concept of birth defects. So the embryonic stage is far more crucial to the development of a baby than the fetal stage, because during these first 2 months, we have all of our heart development, our limb development, our sense development. We do continue to have reproductive and nervous development throughout the pregnancy, but if we're looking at what kinds of medications, exposures, illicit substances could have an effect on the pregnancy, we're really thinking about those first 2 months, which is unfortunate because it may take a while for a woman to know she's pregnant if she's not intending on having a child. So if a woman does not know she's pregnant, we really have to look at the idea of birth defects specific to whatever exposure she's had.